Topic: human excretory system. Now, human urinary system it is composed of a pair of kidneys which are bean shaped, pair of ureter, which is arising each from one kidney, a urinary bladder, and a urethra. Now, where are the kidneys located? Location is kidneys are always located in the abdominal cavity. In the abdominal cavity, one on either side of the vertebral column, at the level between the 12th thoracic vertebra and second or third lumbar vertebra. Now what are these? These are the positions of thoracic as well as lumbar vertebra in the vertebral column where kidneys are located. And it is attached to the dorsal wall because it is not on the ventral side. Kidneys are always located on the dorsal side, located or attached to the dorsal inner wall of the abdominal cavity. This is the proper location. Now, what does the structure of kidney look like? Each kidney, it's a bean shaped in structure. Color reddish brown, like the kidney beans, rajma beans we have seen. Reddish brown, and about 10 to 12 centimeter in length and 5 to 7 centimeter in width. These are the dimensions, and obviously there is thickness as well, and thickness is about 2 to 3 centimeter in thickness. What's its weight? 150 gram. Okay, now how does it look like? The entire urinary system arrangement. We say pair of kidneys on either side. And from this notch known as hilum arises the ureter, which is generally opening up like this. A pair of renal aorta we have to show like this. This is artery, and another like this. Okay, on the top of each kidney, what is located? Adrenal gland. Now, these are the adrenal glands. This is the cortex region of the kidney, the one that we have darkened. And the inner one is, this one is the medulla region of kidney. Then is the vena cava that is entering into it, what is renal artery and renal vein. Urinary bladder, urethra, and these ones long tubules are ureter. Clear? This is the system of human urinary pathway. Clear? Moving to the system in detail. If you can see here towards the center of the inner concave surface, there is a notch. This notch is basically ऐसे होता है, ऐसे curve. ठीक है, इसको हम बोलते हैं हाइलम. What do we call it? Notch is known as hilum. Why? Because this is the passage for ureter, this is the passage for renal vein as well as the renal artery. Now inner to the phylum, at this hilum region, is a funnel shaped space called the renal pelvis with projections. I am drawing that structure now. This is the structure that we are talking about in this. These are the projections. Alright. So inner to the hilum region is a funnel shaped space called the renal pelvis with projections called calyces. These projections are known as calyces. So many in number, so calyces, otherwise singular is known as CALYX, calyx. Alright, and this is the renal capsule. This region where it extends this is the pelvis region. This one. From here the ureter will come out, and from here is the renal artery and renal vein will move out. So we can draw it as well. This way renal artery and renal vein will move out. This region, which is on the outer side, we call it cortex. Renal cortex, and these projections are further have pyramids onto it. These are medullary pyramids which are projected here. In between you can see these columns. This is one column, second column, third one, fourth and fifth. Alright, these are the medullary pyramids. And these are the columns, and these are known as columns of Bertini. Name is bit indifferent. Bertini, B-E-R-T-I-N-I, columns of Bertini. This is the structure of a kidney when we are viewing it or observing it in a longitudinal manner. So LS of kidney, which shows the pelvis region, the cortical region. Pelvis give rise to these extensions known as calyces. Singular calyx, which has medullary pyramids onto it. These medullary pyramids have certain columns in between, and these columns are known as columns of Bertini. I have drawn it with an arrow. Otherwise, these are only the leftover space. That means the white space which is left in between. Clear? This hilum region will show that ureter, renal artery, renal vein all will come out or project out from here. Now coming to the structure of a nephron. What is structure of a nephron composed of? Basically, there are two types of nephrons on the basis of their position in the kidney. One is juxtamedullary nephron. Another one is cortical nephron. As the name suggests, juxtamedullary, when the loop of Henle it dips into the medulla region. Alright, when the loop of Henle it dips into medullary region or medulla region, then it is known as juxtamedullary. Cortical means when the loop of Henle it remains restricted only in the cortical region. We are not talking about the nephron, we are talking about the loop of Henle of the nephron. It's not about the location of the nephron. All the nephrons are located here only, इस area में, maximum, right? But when the loop of Henle dips into the medulla, deep into the medulla, we call it juxtamedullary, otherwise we call them cortical nephrons, right? Now, parts of a nephron. It is mainly divided into glomerulus and renal tubule. Clear? Glomerulus and renal tubule. Now the detail of glomerulus as well as the renal tubule. Glomerulus. It is basically tuft of कैपिलरीज. मतलब इसके अंदर बहुत सारी कैपिलरीज होती हैं. किस चीज़ की बनी हुई कैपिलरीज होती हैं? ऑफ एफरेंट आर्ट्रियोल. From where this afferent arteriole is coming? This is coming from the renal artery. And this is the final, we can say, the finest ever branch of renal artery. Right, finest branch. Then, blood from the glomerulus is carried away by the efferent arteriole. This one brings in blood and there is efferent arteriole. Its role is to carry away the blood from the glomerulus. Then this efferent arteriole, it also forms a finer network known as peritubular capillary around the renal tubule. When it forms a fine network, that network is peritubular network. It surrounds the renal tubule. ऐसा क्यों होता है? One is there forming the tuft of capillaries in the glomerulus itself because it can filter the blood. Another one carries away the blood from the glomerulus. It forms a peritubular network also around the renal tubule so that whatever blood is being filtered out and is considered as now impure along with the unabsorbed material by the tubules is sent to this finer branch and is carried away to the heart. The minute form of this only is known as U-shaped vasa recta. So the efferent artery or efferent arteriole also transforms into a U-shaped vasa recta. That is also which runs parallel to the loop of Henle for the transport of blood to the body. Which blood? Impure blood. Okay. Now next is the renal tubule. What is renal tubule? The first one is Bowman's capsule. Bowman's capsule. The renal tubule is basically closed at the proximal end. Here it is expanded, curved to form a double-walled cup-shaped structure called the Bowman's capsule. अगर आप इसको देखोगे तो ये स्ट्रक्चर थोड़ा सा ना ऐसे नज़र आता है. And over here we say that a tuft of afferent artery has entered, and when it enters it will automatically being taken up by another tuft and it moves out. ठीक है, तो ये एंटर करने के लिए, ये एग्जिट करने के लिए. And this entire structure, this one, this cup shaped structure, it is known as Bowman's capsule. One cell thick so as to show absorption of certain materials, whatever comes along with the blood, right? It is closed at the proximity and is expanded and curved inward, forms a double-walled cup-shaped structure. This is known as Bowman's capsule, which encloses the glomerulus. Now this one in a one is the glomerulus. Okay, now this Bowman's capsule and glomerulus together, this is known as Malpighian body. Malpighian body. Clear? Moving to this structure in detail. Now the lumen of this capsule, it continues with the narrow lumen of the entire tubule. How? That I'll show you in the structure. In this structure, we start from here and we form a tubular network of renal tubule showing various paths where and how it is entangled. This is the afferent artery, obviously, which will bring in the blood. And it forms a mesh-like network or the structure here. And the one that we have drawn is the afferent. Now what the afferent will do, this afferent from here remains continue with the entire length of this structure. ठीक है, ये पूरा इसके साथ साथ घूमता है. Why? Because यूरिन फिल्ट्रेशन, यूरिन एब्जॉर्प्शन, रीएब्जॉर्प्शन, इन सब का पर्पस होता है कि ये वाली पूरी की पूरी जो आर्टरी साथ साथ में इसके घूम रही है, जो भी अनएब्जॉर्ब मटेरियल है, जो भी ब्लड फिल्टर आउट करता है फ्रॉम दीज ट्यूब्यूल्स, दैट इज एब्जॉर्ब्ड हियर. Ions, these are absorbed here. Glucose, amino acids, sodium, potassium, bicarbonate, hydrogen ion, all these along with the blood are carried here. Okay, so this will bring in and that will take out. What are the different parts? This we are clear with. This is Bowman's capsule. This is glomerulus. Here it shows a network of small cells and we call these small cells as podocytes. क्या बोलते हैं? Podocytes. Why? The best or the finest filtering unit होता है. Its diameter is so less that easily everything can be filtered out from the blood, avoiding all the major particles like proteins. Proteins बहुत बड़े होते हैं, carbohydrates बहुत बड़े होते हैं. No. Okay, amino acids, glucose, sodium, potassium, chloride, iron, urea, whatever is in the simple form and inorganic form and in a simple absorbable form can be easily filtered out through these podocytes, right? Then this part which is near the Bowman's capsule, we call it PCT, proximal, right? And this part which is far away from that, we call it DCT, distal convoluted tubule. So CT stands for convoluted tubule. If it is PCT, proximal. If it is DCT, distal. P stands for proximal, D stands for distal. In respect to the position of this Malpighian body, then this U-shaped is loop of Henle, and this projection that we have already discussed, it is the efferent arteriole which takes the U-shaped turn and runs all along the entire length of loop of Henle is vasa recta. Clear? Here, this one is the collecting duct where the entire blood has to be poured. Okay, and this glomerulus and Bowman's capsule, it is known as Malpighian body. Now, we say the lumen of the capsule is continuous with the narrow lumen of the entire tube. Clear? Coming to PCT, the first one. It's PCT. PCT, it forms a highly convoluted proximal cuboidal tube. Okay, it has brush bordered cuboidal epithelial cells. What for? These cells lines the lumen of PCT. Why? So that to carry away all the contents. Then is loop of Henle. From where does it arise? From the end of PCT. It's a continuous structure. End of PCT will give rise to the loop of Henle. Inverted hairpin-like structure. Okay. It has a descending limb. This one is the descending and this one is the ascending limb. Clear? So, this structure, U-shaped structure, is the loop of Henle. It starts from the proximal and it goes or reaches there near the distal, meaning it ends at the beginning of the distal tubule, at the starting of distal tubule. Then DCT, distal convoluted tubule. This ascending limb of this DCT, it becomes highly coiled and convoluted. Highly coiled and convoluted structure. It continues as a straight collecting tube, this area, straight collecting tube which joins the collecting duct. What is collecting duct? This is collecting duct. This collecting duct, it receives the collecting tubules of various nephrons. See, check here. These are the collecting tubules of various nephrons, different nephrons which are lying here and there, and this is a common duct. And these converge, run through the renal pyramids, open into the pelvis. Pelvis, you remember? The structure from where the calyces arise, that is pelvis. It opens there and pours everything into the ureter, from where it is carried into the urinary bladder, and finally everything is expelled out through the urethra when the urge comes. So the fourth one is collecting tubule or the collecting duct. This is the collecting duct which collects the tubules of all the various nephrons or different nephrons. This is about the structure of a nephron. Now, whenever we cover with the structure of nephron, the next thing that comes to our mind is how the urine formation takes place. Moving to the next video for the urine formation.